I don't hear any sound on the YouTube.
Hello everyone, welcome to Astro Stream. I hope you can hear us fine and you can see me as well. And today we have Reut with us joining for our introduction to astrobiology course. And I hope you all are excited to hear from her. We have had an introductory session from her beforehand back on 16th of August, I guess, so you can find it on our YouTube. And she's going to be with us every Sunday to talk about astrobiology. If you want a certificate for this course, there is a form link given in our description. You can fill it up, and after all these sessions are done, we will send you your own certificate. Of course, there will be a small quiz just to know how sincerely you have attended this session, because for our previous Astro stream with Ellen Glad, if you have seen it, we have had thousands of people watching that stream, and we are still processing the certificate. So I just want to thank you for your patience, and I hope we'll be able to send them all out next week. So without further ado, we are going to start with Reut, and make sure that you stick until the end of this session for the next announcements. And if you have any questions, do post in the live chat, Reut, and I can see it, and we are going to answer your questions there as well. So now off to you, Reut.
Hello. Thank you very much. Very happy to be here again. It's good to see you, everybody. It's good to see everybody coming in and starting to watch us. I'm going to have a very quick recap of what happened in the past episode, because I think some of you are new participants. So we were starting to talk about astrobiology, which is a multidisciplinary field, and it has many questions to answer about the origin of life on planet Earth and in other places in the universe. How did life evolve? What is a habitable zone where life can survive, and how life is distributed in the universe? What are extremophiles? What kind of technologies we use today to find extremophiles and to find life on planet Earth and also elsewhere? So we're going to talk about all these topics, but today we're going to focus on one of these topics as promised. So I'm an astrobiology. I'm also working at 2 NGOs at the moment, volunteering here for the Israeli Mars Society and for DMARS, doing in analog missions. I'm a research fellow at the Dead Sea in Arava Science Center, and I deal with cross-contaminations issues in analog missions. I'm a public speaker, and I'm a good friend of Shweta, which is why I'm here. So without further ado, I'm going to share my screen with you, and we're going to start the presentation I prepared for you. Again, stick to the end. You can get a lot from us answering your questions. Each of us are experts in our own field, and I would be delighted to talk to you afterwards. And also, I would like to add that in the next session, not this one, but in the next session, I'm going to give you a riddle. So if you want to come next time also and enjoy my riddles, it's a very geeky astrobiology riddle type of thing. So come back next time as well. So this is a general introduction. So astrobiology in the universe. I'm very happy to be here. These are the 5 topics. And today, after I've explained again the different issues that we deal with astrobiology, today we're going to talk about the beginning of life. The beginning of life is a super interesting topic. We don't have finite answers for anything, so you can ask whatever you want. Everything is open for discussion. So basically today I'm going to focus on 3 items. I'm going to focus on the origin of life as we understand it today, following decades of research. I'm going to talk a little bit about our atomic and organic elements, what is in the universe, what is on planet Earth, what are the entire biological system. We're going to, through this, I hope you start to understand a bit more about what is life, where does it go, how do we define life, and so on and so forth. What are the basic components of a life form on planet Earth, and what we think would be a components relevant for life elsewhere in the universe. I'm going to talk a lot today about the different theories about how life originated on planet Earth. So here it's a big circle called primordial soup, but basically there are 5 different theories today and how life has began on planet Earth. We're going to go through them. It's going to be a little bit scientific, but I think you're going to be very successful in understanding what I'm saying. And as I said, at the end, ask your questions, and I would be happy to answer. The basic question, which I've also asked it last time, is why search for life? Why search for life? For me, it's such a basic question that I really need to pause from time to time and look again at my answers. It's so obvious for me why to search for life. People tell me different issues they have with life and how, civilizations on planet Earth have been using plants very successfully, how they came to work with the nature around them. So we are a very successful life form on planet Earth, and we are really good at reaching out, exploring our environments, our physical environments, and finding different life forms. We like to search for life because we are surrounded by different life forms, and we are using them in our everyday lifestyles, choices that we do here in our lives on planet Earth. We have, for instance, microorganisms which help us with our food and our beverages, wine, beer, cakes, bread. You use yeast, you use lactobacillus. We use fungus, and we use different type of bacteria to produce medicine. Important antibiotics come only from medicine and helps us to alleviate disease and infections. And we're using, again, a really large section in bacteria, which is called secondary metabolites, in which we use them to produce many things that some of you might not be aware. But for instance, the pigments that help to screen UV, for instance. And astaxanthin is it's a pigment that an antioxidant basically, that we harness and we produce from red algae. We are able to use it as an antioxidant, and it's now sold in basic pharmacies and everywhere as sort of like an eye health because it's an antioxidant. And there's plenty more where that comes from. Our gut microbiota is very much dependent on the composition of its microbial population there. So basically, life is not only around us, it's inside us. We use it every day, whether it's on our skin, whether it's in our food. And you can say that we have a very rich and deep interactions with microorganisms and with life forms on this planet. So searching life elsewhere is something that we almost do offhand. We're curious about it. We know how to use it. We have centuries of knowledge on how to do these things. So when did life begin? So what we know now from decades of research is that we think that life has arose. And when I say life, I mean a simple cell or a few cells together. We think life as a community, as a microorganism, has started about 3.5 to 4 billion years ago as a microorganism group. What you see on the left side of the screen, these squiggly lines in a rock, which was in a beautiful image taken by Jessica Kofi. It's a very ancient rock bed coming from the Pilbara region, in Western Australia. And those squiggly lines were not formed just by the action of the ancient waves of the ancient ocean that was there 3.43 billion years ago. And this is how old this rock is. But they were formed together with the waves, together with microbial mats in a format that we now call stromatolites. And stromatolites, it's a word from Latin. Stroma means spread, and lit means stone, from litus. And so what we usually find is that in certain areas in the world, it could be in Canada and in China and in other places, like in Australia, but places which are very old, in the rock bed, we start finding these images, these shapes in the rock. And geologists about 250 years ago decided that these shapes do not come just from physical and chemical degradation and conditions at the time that the rock was formed, but also from microbial components. So these are microbial mats. They form layers upon layers upon layers upon layers of rocks for thousands and thousands of years. And the beauty is that in Shark Bay, where you see the red arrow on Australia, that's actually where I did my PhD. And in Shark Bay, you can find stromatolites that are doing the same thing today. So you can see the process happening before your eyes. And it's an amazing place to go to. And it's an amazing process to understand the component of the stromatolites within their geological setup. It's very, very interesting. And we have a few more, I would say, evidence from about close to 4 billion years, about microfossils, and they're very, very rare and very, very delicate, but we do have some evidence. So we think that life on planet Earth started about 3.6 to almost 4 billion years ago. And when you take into consideration that the planet is just 4.5 billion years ago, then what we're saying is that life has arisen probably within 500 million years on planet Earth. And that means that if we had Mars, for instance, and Mars in its first billion years was similar to planet Earth, had an atmosphere, had oceans, then there's a good chance that if we found here evidence of stromatolites, we actually think that there might be a good analogy on it on Mars in very ancient rock beds. Not today, but in the past. So this is about the atomic elements that I was talking about. Sponge, okay? Just say it out loud. Nobody else hears you. I'm going to say it again. Sponge. Sponge basically represents the elements sulfur, phosphate, oxygen, nitrogen, carbon, and hydrogen. And 99% of all living mass on planet Earth has these elements. And once in their basic composition, so nitrogen is in our nucleotides, in our proteins, carbon is in everywhere, in sugar, in membranes, in proteins as well. Oxygen, as you know, part of water, H2O, we breathe it in, it goes out again. Phosphate is a good part of our nucleotide, of our genetic code. It's also part of an energy molecule called ATP. And sulfur is a super important element in our enzyme. It appears in many, many places. And so what's interesting about these elements, which every living thing on planet Earth has them, is that they are very common in the universe as well. They're in the top 10 common elements in the universe, which is why when people ask me, oh, what about, like gold-based alien life forms? And I'm like, well, really depends. We don't know on any place in the universe where gold is so common as hydrogen. It just doesn't happen in our physical universe. Therefore, the issue of arising life from gold, it has a very, very low, really low, tiny probability to it. As a scientist, I cannot say an absolute no. That's not my style. But as you can see, all living mass on Earth are using common elements which are common in our universe, common in the Milky Way, in our galaxy, and common on planet Earth and in the solar system. So there's a correlation there. And you need to remember these things when we go through all our different topics about, okay, so how did life arose? What is a life form? How did they use these different elements? What did they build with it? So as we talk about what is a life form, I want to show you this beautiful video by this artist and chemist called Emmanuel Foranzier. And what they're showing here is something that I like to point out, is that people think that life forms grow, that growth and reproduction is an essential criteria for life forms. And what this beautiful video shows, and I don't know if you can hear the music with it, it's a beautiful piano segment. It shows how these metal crystals, this is copper, this is silver, this is lead, they grow in different ways, and none of them are biological, right? They're all abiotic. So I'm going to give you a minute or two to look at it and just enjoy this beautiful, beautiful video clip. And so when someone tells you, oh, I've seen something that grows or something that looks like it's reproducing, that's a very ethnocentric point of view. And it's not what we describe today as something that is vital to say this is a life form or not. So I just wanted to point that out. We know a few other processes in geology and in minerals in which crystals or semi-crystals grow and become bigger. And some of them even look like trees, but has nothing to do with a biological process behind it. It has everything to do with atomic and electronic and electrons transfer, as this beautiful video as just shown. So when we're talking about the origin of life, there are, like I said, 1, 2, 3, 4, 5 topics I'm going to talk today, which about the origin of life. So one of them is called the primordial soup. One of them is called the RNA world. Another is panspermia. The fourth is a general topic called autocatalytic sets. And my favorite, not exactly my favorite, but one of my favorites is lipids first. So Charles Darwin is one of the biggest Western naturalist, scientist, and philosopher. And he already in the 19th century said this beautiful paragraph. And he said, but if, and oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex exchanges. And basically he pointed out that the logic behind life forms that need protein or enzyme. And he says, okay, we need certain nutrients, we need certain elements. He's talking about ammonia and phosphoric salts here. And then he says, okay, we need a source of energy. And he talks about light, heat, or electricity. And so following several, I would say 100 years, almost 100 years later, two brilliant scientists from the University of Chicago and later on from the University of California in San Diego, they are called Harold Urey and Stanley Wiener. They did the very famous experiment, which is taught in every biological one-on-one, and it's called the Urey-Miller experiment. And they basically recreated part what Darwin meant and part what they knew about the early atmosphere of planet Earth. And this was in order to understand how amino acids are formed, when amino acids are the basic building blocks of proteins. And in our planet, we know on 22 different amino acids, and everybody is using 20 of those. Two of those, the additional twos are very unique variants, which are not common across our biosphere, but 20 amino acids are. And what they did was a very clever thing. Basically, what they did is they boiled water, they made it into vapor. That vapor went along into a new vessel. This new vessel had an atmospheric composition was that in 1952 was considered the early atmosphere of planet Earth. And that atmosphere included carbon dioxide that we have today. It included nitrogen gas that we have today. It included ammonia, which we don't really have. It included hydrogen gas, which we have very little, if nothing, only really down in swamps and things like that. Methane, same goes. We don't have it in our atmosphere at the moment. Water, we do have quite a lot of water vapor. And carbon monoxide, which we know as now is something that happens mainly in big cities due to pollution. And what they next did is that those water vapors going into this vessel with this atmospheric, basically what they did is that they put lightning, an electricity source, right, an energy source, and they did it 7 days and 7 nights in a row. And they gave energy to the molecules that came into this. And what happened afterwards is that the concentrate, the results from that experiment went down here into this valve that they were able to open. And then they could analyze all that organic compound, which is basically looked like a black goo, basically, and check what has formed there after all these, electricity and lightning and vapor and the original atmospheric. And what they have found out is that they were able to produce a limited set of the amino acid that we now know are on planet Earth. And first of all, they said, oh, our experiment failed, because they were expecting to find the 20, the full diversity of all the amino acids that we know today that will build proteins. But even what they found was quite amazing, because that provided a proof that we can create amino acids from an abiotic process, from a physical chemical process, and that's that. And following their experiment, a lot of people did variants. They changed the different atmospheric condition, because now we know there wasn't ammonia much in the early atmosphere. There was carbon dioxide, there was hydrogen sulfide, there were volcanic ashes. So we know there's a lot different type of nutrients, and people have repeated this experiment in hundreds of variation, and eventually we now know that they produce almost all the amino acids that we're looking for that we have now on planet Earth. So it's a biotic generation of these amino acids. And when we started doing proper astronomy as well, we were able to detect these amino acids on meteorites, in different planets, in different stars even. And what we realized is that amino acid are very common in our universe, and amino acid is building blocks of proteins. You can find them from out of our world as well, and they were able to form in planet Earth. Interesting topic here is about chirality, which is a asymmetric kind of way of molecules. Basically, what the picture here of your two left-handed, it's called a chirality, and these two represent the bulbs inside, basically represent a chemical formula of one amino acid. This is the basic structure of an amino acid. And what we found out is that some amino acids are left-handed and some are right-handed. And in this issue of chirality means that both molecules function exactly the same in the real world, but they are built differently, just like you cannot superimpose one upon the other. And it has some physical meaning, which means, the physical meaning has to do with how the polarized light twist when it goes through a right-handed and versus a left-handed. And it's not a big issue. I don't want you to remember the chirality right now. But the primordial soup was the first time when something that Darwin talked about in the former century, in the 19th century, we were able as scientists to actually put some evidence to it in the 20th century. And it gave us a big boost in understanding how the beginning of life might have been. But I'm going all through this about life without actually defining what a life is, what a life form is. And that's because I don't want to go into the metaphysical issues of what is a life, because this is a scientific lecture. But I do want you to understand that today we know what are the cell components and what are the mechanisms in which life forms on planet Earth, how they evolved. And I'm not going to go into the differences between the virus and the bacteria and the eukaryotes and archaea. Not going to go into those details. I'm going to try and explain to you very briefly how a cell component on planet Earth looks like. Basically, it has a membrane, different types of membranes. Some of them have silica, some of them don't. Some of them have sulfate, some of them don't. The membranes are an envelope. They have canals inside. Some of them have like small rotors, actually, to pump things out or take things in. It's an active membrane. It's not a completely, absolutely shield from the environment. Inside the cell, we have something which is called the DNA, deoxynucleotide acid. That's the genetic code. It codes. It's like a program in a computer. It codes everything the computer knows to do, or our bodies and different bodies can do. It is combined from 4 repeating bases, which are called adenine, guanine, cytosine, and thymine. And basically, these are all are backboned. They're one attached to another, and they're also combined like into a helical. So some viruses work with only one string of DNA. Most of our cells have, of course, double DNA strings inside, and so that is the code with which we work with. That's the DNA. It's a very stable molecule, and we have now pictures with electron microscopy at how it looks inside a cell, and you can look it up in the internet. However, what really works in the body are the proteins. So how do you get from the code, which sits very nicely inside the cell, to all these proteins and different metabolites and molecules? How do you do that? So this is now known as the dogma. What you see here on the left side with all these errors and everything is the dogma on how a cell works today, how a life forms on planet Earth, each and every cell works. Each bacteria, archaea, even viruses usually work around these, have a variation on this issue. So basically what we have is from the DNA, we have another nucleotide base molecule, which is called RNA, ribonucleotide acid. And what we see here with the M, it stands for messenger. What these messenger RNAs do, they duplicate what they find on the code, like in specific segments on the DNA, and they bring it to the ribosome. A ribosome is a big machine, is a big protein in every cell, okay? In every cell we find this big protein, and we have Professor Ada Yonat from the Weizmann Institute to thank for elucidating its structure, its 3D structure, and getting a Nobel Prize for it. Good for Ada. And the mRNA then works with the ribosome in order to bring into the ribosome each amino acid in a certain sequence. Once that sequence is completed, you get a protein, okay? This is an oversimplified way of talking about it, but it's important to understand that we have these different mechanisms. We have the genetic code, we have the messenger that goes between the genetic code and into the ribosome, and then we have all these plethora, like thousands and thousands of protein working. What are they doing? They're shifting molecules, they're shifting electrons, they're changing things between the membrane and the inner cell, between the DNA and the mRNA. They're everywhere. So it's a very complex system, and people are starting to think, wait a second, so what was the first molecule? Was it the functional one, the protein? Was it the genetic code? But the genetic code is pretty passive. Like, it's not doing anything in the cell. So it cannot be, if you're a cell and you need to survive with nutrients that are outside of your membrane, then how do you do that if you're completely passive? No, you need the messenger. But so people are trying to figure out which of these different components came first or did some other functions. And this is the interesting topic about the beginning of life and the original life of planet Earth, how these things came into being. So if we're doing just a mid-summary, and you're doing very well so far, okay? Very well, stay put, you're doing great. This is a tough lecture, I know. But we talked so far, we talked about why it's good to search for life forms, because it helps us to be better, because it's part of our nature anyhow. I talked about the fact that life has probably arose on planet Earth about 3.5 to almost 4 billion years ago. I was talking about the common elements in atom that we have in our 99% of our masses on Earth, which is correlating to the 10 most common elements in the universe, which is sponge. Remember that, sponge. I was talking a little bit about the Urey-Miller experiment, which was fascinating and has like a whole Wikipedia thing on it. And it's really, really an interesting topic, and all the different variations that people, in order to understand how abiogenesis, how from an abiotic, physical, chemical, you get a building block for life. And that building block is an amino acid. I talked a little bit about chirality and amino acids. They're interesting topics to talk about. They build your proteins through different stages of structure until the protein is a beautiful 3D structure that has many functions to do within a cell. And I talked a little bit about the basic cell components. So we talked about the DNA, the genetic code, the mRNA, which is sort of like the program that runs. And then we had the proteins, which, I don't know, maybe you can say it's in electrons or something in a computer. But basically proteins are the horses. They're the ones that are working in the cells. And each cell has thousands and thousands of protein. Even viruses has between tens to hundreds of proteins that work for them as well, even though they're so small. So when I'm moving forward, I now want to talk about panspermia. And panspermia is a super interesting topic that has been around for decades, and we're not finding a lot of proof for it. And let me explain what it means. Panspermia means that life on Earth, or even building blocks of life on Earth, have arrived from outer space. Simple as that. So there was a meteorite coming from Mars or from Titan or from I don't know where, some asteroid that came through the solar system and went elsewhere, and it smashed on planet Earth, and inside that piece of meteorite was the golden cell. Maybe there was already a living thing, and it found a really nice pond on planet Earth. Maybe there was amino acid we know for sure came from outer space as well, but maybe other building blocks. Maybe it was protein, maybe it was DNA, who knows? So that's panspermia. What's happening with panspermia is that it's very hard to prove when you think about it, and it doesn't provide you really with the real answer to the interesting question is, okay, so it came from a different planet. It doesn't explain to you how it started on that planet. It doesn't really tell you how life has started actually. But it's an interesting theory about the transport of life in an interstellar space. And what we did find along in the last, I would say, 40 to 50 years is that you can find microorganisms that survive in space conditions. We find bacillus and we find lichens and we find tardigrades that even if you expose them to the vacuum of space, they don't die. They maybe go into hibernation, maybe they form a spore, and they are very well held in it, but they don't die. After you put them back inside the spaceship or you take them back to planet Earth under control condition, you can see that they're working just fine. So that's panspermia for you. It's a lovely topic. Now, what do you see on the right side is an amazing photograph, a microscopic photograph of a meteorite that was found in Antarctica in Allen Hills in 1984. And in 1996, there was a really big science article coming out of it. And the author claimed that you see this little thing in the middle that looks a little bit like a worm or something like that. That's actually a microfossil on a Martian meteorite that came from Mars. And that might be evidence for Martian life forms. So this caused a big debate and discussions for decades. What happened when this was published is that aside from how it looked, okay, nobody could prove that it had DNA, that it had any protein, that any kind of a biomarker that comes from a biological activity has been assigned to this piece in the meteorite. But what else was found out is that when you look at this meteorite, this piece of little thing is actually on the outside of the meteorite, which means if this meteorite has landed 50,000 years ago in Antarctica, and we know about winds in Antarctica, and we know that about other things that go through the air, there is that tiny chance that within 50,000 years old, right, something actually landed in that field. Something from Earth, not from Mars. Something from Earth has landed on that meteorite. So from then on, this is like a cornerstone for how you deal with meteorites and how do you deal with potential microfossils from other planets and from outer space, basically. So today the consensus is that this is not a microfossil from Mars. The meteor is a Martian meteorite because we have found it out through isotopic analysis in the mineral composition. So we're sure that it did actually land it from Mars. But the thing around it, no, nothing to do with a life form from Mars. And I'm looking forward to the day when Perseverance or other rovers or other robotic missions or perhaps a human eye would look at a sample from Mars and go, that's a life form. And it's a life form because it ticked a lot of boxes, not just one, not just because it looks like it, but also all the different other boxes. That will be something amazing, absolutely amazing. So moving forward, let's talk about the RNA world. So remember that we talked about how a basic cell works, and we said it's a complex system. There's so many agents working on it. So everybody in science were like looking for a simpler solution, something that perhaps could be a DNA, have a code attached to it, but also perhaps will be a little bit like a protein, like a functioning 3D molecule that goes there and goes over there and, pull this and, passes around in electrons and things like that. And what people are really, really they like the RNA molecule. On the right side of the screen, you see how it's a 2D graph, a representation of a 3D molecule. RNAs are nothing like DNAs. They're flexible. There's like hundreds types of RNA. Some are long, some are short. They're able to change their configuration, and they talk with genetic code. So basically, and sometimes you have proteins which sort of like translate the RNA themselves. So people are thinking, wait, I have here one molecule, which is an RNA, and it does both. It has the genetic code, and it's able to function in enzyme. So people are really thinking that perhaps first there was an RNA world, and later you had this speciation between something which is a genetic code, stable, doesn't work so much, and then you have enzymes which are much more flexible and can catalyze reaction, cause them to happen much more quickly than perhaps an RNA molecule sitting in one portion of the cell. The main issue with RNA is that it's a very delicate molecule. It's destroyed fairly easy. inside a cell. It's short-lived. DNA, for instance, you can go to burial sites from the Paleolithic time frame, find a bone of a human being from about 200,000 years old, and you can check its DNA with current technologies. Yes, you're going to have to be very careful and gentle, but the DNA would have survived UV and radiation and pressure and changing temperature and all those things. It's a super stable molecule. RNA, not so much. So people are not sure how that molecule was able to work till today for 4 billion years. Nobody's actually really sure about RNA. Now with the DNA, it's an interesting thing because people are saying, wait a second. So DNA now is very stable, but perhaps in the past, and we have now synthetic biologists who were able to synthesize this PNA, which is a peptide nucleic acid, and 2-aminoethyl glycan, AEG for sure, which basically forms a helix, a double helix, just like our DNA, with different type of angles and so on and so forth, and it can work as a DNA. You need different RNA to translate from it, and it needs other conditions, but basically it has almost the same features as our DNA. One of the most interesting articles came out in 2012 when people actually found the AAG, this PNA style thing. They found it in living today cyanobacteria. Cyanobacteria are bacteria which in my PhD, I investigated. They're photosynthetic. They're the first photosynthetic bacteria in the world. They are the reason why we're all breathing oxygen. They have arrived in our world probably about 3 billion years ago. And the fact that they live till today is a testimony to how resilient as a group they are through all the different, through all the different evolutionary disasters that we have here. But cyanobacteria today produces this AEG, and we can find it in different groups of cyanobacteria across the world. So we're very happy to find this finding. So perhaps DNA didn't start as DNA, it started as short peptides that later on specialized as DNA. So that's a theory. We don't know for sure anything. When we want to talk about the lipids first, lipids is a completely different component. We're talking here about the membrane. All membranes on planet Earth are formed from something which is called a lipid. And you can see it here as like a stick with 2 red oxygen atoms at the head of it, at the head of the tail. So basically the head of the tail is called hydrophilic. It loves water, and that's the inside. It comes on the inside of the membrane, and the outside is called a hydrophobic tail. And a hydrophobic means that it hates water. So basically, if you put this as an array, one next to another in the same vector, in the same position, you're going to get an inside which hates, okay, which basically hates water. And on the outside, you're going to have something which is happy to be soluble in water, and so on and so forth. And what you see here on the membrane is that you see that there are like 2 sides of this. So you have the inside, which likes water, which has it's a tail with 2 entrants. But in the midterm of it, in the middle of it, you have all these hydrophobic tails, which exclude water outside of the functioning quorum or the functioning hole inside this cell. And what the beautiful thing with this lipid world scenario is that we now have a lot of evidence saying that micelle, which is the middle, I would say like the middle form between just a lipid, one lipid or a few lipids in between a fully formed membrane. So these micelles and membranes can form aphysically and abiotic completely. They don't need any more. They don't need energy around them. They don't need heat around them. They don't need an energy source around them. Like I said, they don't need any nutrient. They're all ready. They come as they go, and they perform, and they make these little balls. And what the beauty of it is that this actually means that if, for instance, we had all these different membranes and we have this micelle, these spheres working around, very easily you can imagine that these spheres are enveloping basically some molecules. Could be an RNA, an early RNA, could be an early DNA. But once you envelope them, you shorten the distances between these molecules, and you allow them to interact and develop and break and send electrons and perhaps have variation. So when a membrane is formed, when an envelope is formed, it's like forming like its own small planet, and allowing for a lot of things to happen inside. And there's a lot of, membrane can form pretty easily according to latest research. And that's called the lipid world scenario about this, life component, which is very important, the membrane. And so it brings us forward to the lovely autocatalytic sets. So autocatalytic sets in the origin of life is one of the latest publications in 2010, but of course there are later than that. But the idea is that once you have a membrane, once you have a set of certain molecules put together, they catalyze their own reaction and create their own molecules. So within this enclosed area, you have your own process of evolution going on. And they duplicate perhaps with mistakes. And all of a sudden, instead of 3 molecules which were pretty much the same and made out of perhaps the same even elements, because you have a membrane which perhaps allows for sulfur to enter or nitrate or something else to enter, not nitrate, but basically phosphorus or anything else to enter, all of a sudden you have your own, game area. And autocatalytic sets, from a mathematical point of view, are a great example of how life could just start very simply, but very quickly become into this very complex set of things once you have them enclosed inside an envelope, which is why it's important to understand how easy or not easy it is to have lipids first. So this is about autocatalytic sets. It starts from an inorganic compound with the abiotic synthesis of organic monomers, for instance, amino acid, as we've seen before, and then you put them into organic monomers. They're starting to be a polymer. There's like a chain or a backbone of more complicated molecules. They're getting a bit bigger, but not too big. And then there's the formation of a pre-cell, a protocell, perhaps an envelope, a micelle around these things. But what happens then is that they start to replicate, to work one upon the other, each one having a discussion with the other molecules within this envelope. And lo and behold, you have this microevolution happening at faster rates. And because that, all of a sudden you get really complex system just out of, probability and how fast, and you give it enough time. That's how things happen. So when I was talking about all these things, I want to let you just a little bit about how the sequence of events, how we think the sequence of events might have happened so far for these really basic components of life. And I know it's a lot to digest, so please feel free to ask questions afterwards, and I would be happy to answer them. We're starting with inorganic chemistry. We now know that just like micelles, like lipids form, and just like amino acids form, we probably have a few more items on our life components which can start with inorganic chemistry, basic physics. Then we have something really simple, like a prebiotic metabolism, which means one molecule affects another, which affects a third, which affects a fourth, and perhaps the fourth affects the first one, and so on and so forth. So you see cycles of metabolism, but they're very simple with very few components. And then we think sometime along that way, you have an RNA world. Once you have an RNA world, all of a sudden you have a genetic code. So the cell all of a sudden remembers, have a memory of how things should work. And there's like a reference code, sort of like a genetic code, but not a full genetic code for how things should be and make it stable, not completely dissolved into the world. From RNA world, because we have this functionality, some of them would become proteins, perhaps. RNA nucleotides talking with some of the amino acid inside this envelope. All of a sudden, you have something which is very functional and helpful for the cell, and you get proteins. And from proteins, we can then go back and make the DNA the beautiful, stable, relatively passive, not completely, relatively passive genetic code that forever and ever keeps a record of all the different functions, all the different proteins that a cell needs in order to survive. This is sort of what we came to in conclusion now in the scientific world. However, we didn't talk about time scale. We have no idea how long how long these envelope progressed, how long was the prebiotic metabolism, How long did it take from RNA world to completely develop a really robust protein population? And where all this happened? I'm talking constantly on these micro things. Did it happen in the sky? Did it happen in the ocean? Did it happen on land? Did it happen in lava tubes? Where exactly in the world? Show me where it happened. How does it happen? Were we able to do this in a lab? Not so much. And also, I would like to point out that there's the issue between linear and parallel processes. So you know how this sentence is now on your screen and it looks like very linear from one thing to another and it's very organized. But one of the things that I found out, and many biologists find it as well, is that evolution likes to work in parallel processes. In some places of the world, it acts 1, 2, 3. In different places of the world, it starts completely different and goes into 5, 6, 7. So I'm going to suggest here that some of these components happen in parallel processes in different locations on planet Earth with very different timescales. And it's 4.5 billion years. So it's a big, big time scale to put all these things into together. And where did life start? That's a really, really good question. And I'm not going to bombard you with all the different things, but today there are two really main points where scientists have suggested. We're not sure of anything about how life has started on planet Earth. And on the left side, you see a black smoker. So we have discovered only in the 1970s that the depth of the ocean, 5,000 meter depth, 7,000 meter depth, even 11 kilometers depth, we're talking about 1,000, one with 3 zeros of atmosphere pressure. It's huge pressure, but we found out that life thrives near black smoker, white smoker, in all these ridges because they have adapted, because those black smokers are basically underwater volcano and there are openings to the earth crust and they put out so much nutrients. So everybody who lives there, they get their iron, they get their sulfur, they get their oxygen, they get their hydrogen, they get so many ammonium, nitrogen, they get all the components we talked about in a very rich environment. It's super hot. So even though it's down below and there's no sunlight, it's super hot. It can be 700 Celsius degrees. It could be even higher. And it has a lot of energy to it. So only in the 1970s, we realized that perhaps we now know that life started in the ocean. So maybe in the ocean, we're talking about these, mid-ocean ridge hydrothermal vent. Super hot, very rich in nutrient, and with alive ecological niches around them. The point is, and Stanley Miller also pointed it out, is that under these pressures and under these temperatures, the basic component of life tend to break up, which means amino acids, proteins don't stay connected in any way. Membranes, they tend to split apart, and so on and so forth. So it's not clear to him how life has begun. When you're thinking of the simple beginnings, this doesn't look like an environment that is very good for something simple to just, fold around something else. It's a very harsh environment for those simple creatures. So we're not sure these are actually where life began, but that's an option that people have been discussing, and I'm bringing it to you. The other thing is, so it's a picture here on the right side from Shark Bay. You see all these different rocks. So they're not really rocks. They're actually microbial mats inside them. If you take a hammer and you punch one of these, what you see inside are layers, and some of those layers are moist, and they're actually different microbial layers. Some of them like the oxygen, some of them don't. Some of them do photosynthesis, the others don't. Multi layers of microbial mats. And these were able to form in a very, very shallow bay, very calm. And people think that at the beginning of planet Earth, they were vast oceans. Perhaps they had a lot of shallow bays or areas, ponds, which got the heat from the sun, had a lot of nutrients. And basically, you started seeing these things happening together because nobody was there to interfere, basically, with the system. It was a very convenient place where all the nutrients were, and not too much, I would say, it wasn't too much aggressive from a thermodynamic point of view. So perhaps places like Shark Bay and other areas in the world were the places of the beginning of life, because we know stromatolites were fairly early on in our planet's geological record. So this has been about where life might have started, either shallow ponds or perhaps hydrothermal vents. So in order to end this, and if you've come so far, thank you so much. I really appreciate it. I know it hasn't been easy. You're doing swell. So today I continue to talk a little bit about how the beginning of life started. So for instance, panspermia, that life has come to planet Earth from outer space, from other meteorites. Perhaps the building blocks have arrived here on an asteroid. We talked a little bit about the RNA world, because the RNA molecule has some components of it that work like a protein, but other than the RNA world, because an RNA molecule basically is part a genetic code and part an enzyme, so it can do both. So perhaps got it.
We got the connection now, I guess. So can you start again, please? Yeah, we are online now. Yeah. I'm sure that it's online now. Yeah.
Right. So hi, everybody. To end and talk a little bit, just finishing this presentation after a few of internet problems and so on and so forth, I continue to talk about these topics about the origin of life. One of them is panspermia, about how life building blocks and perhaps life itself in its simple form has arrived from outer space, maybe on a meteorite shower or an asteroid or anything like that. RNA world as a really good candidate for the beginning of life, because RNA is a molecule that it's half enzyme, works as an enzyme, can work in a 3D environment very well, but also has a genetic code in it. It has the same nucleotide, almost the same nucleotide bases such as DNA. I've talked about the lipids first, how like a micelle would be formed, an envelope would be formed around any type of molecules, and it would form abiotically very easily when it has the right physical condition, and will provide like a safe haven, like a sphere for whatever molecules are happening inside. Briefly, I talked about autocatalytic sets. If you have a mathematical inclination, I suggest you read further. It has a lot of mathematics behind it, quite fascinating. And it talks about how in a certain environment, a set of complex molecules can actually push forward and multiply and reproduce and produce also a diversity molecule population, and it's very interesting. Where life has begun, we think that it might have started at hydrothermal vents. We're not sure. We think it probably would have started in a shallow pond, something like stromatolites from Shark Bay or stromatolites elsewhere in the world, and that's how we think life perhaps has started. First as the building blocks, then as a simple cell, and then as multiple cell, like having a microbial population actually working together and using the nutrients and the physical condition to survive. Just additional goodies that I wanted to give here, and I will give probably in every of my lectures, that a recommendation on really, really good books that deal with the issue of life in the universe. So one is by Carl Sagan, who has fantastic books in general. And this one is about the vision of the human future in space, pale blue dot, not A, but it's just pale blue dot. And it's, of course, available in Amazon and perhaps also in an audible version. And the recent one is called Alien Oceans by Kevin Han, The Search for Life in the Depths of Space. And I highly recommend that one as well, because it talks about the oceans that we now know exist in Titan and in Europa and Enceladus and in other places. And it can give you a really good overview and also an in-depth, fascinating view on how science goes and research these places and how life can survive there. So without further ado, I want to say thank you very much. And we're opening for questions, I guess.
Okay, can you stop sharing your screen? Now I can have only you on the screen then to answer the questions. Okay, so we have a few questions by Adarsh Kumar. Let's start with those. He's asking, what do we currently know about the possibility of life forms on Europa and Enceladus?
Okay, so Enceladus, in both places, we don't know about life. We do know there's quite a lot of organic material and there's water in two different settings. So Enceladus, we discussed with Cassini, I think it was the Cassini spacecraft towards Saturn that discovered that there are plumes coming out of Enceladus. And when we analyze those plumes, we realize there's molecules of water in it, there's nitrogen-based and carbon-based molecules. So that's super interesting. And the interesting thing is that also if you have such a strong geyser taking out from the planet's atmosphere into space, something which we don't know from planet Earth, but if we do have that, then it's a fascinating place. It means there's a lot of energy, there's a lot of dynamics going on, and therefore, if you have the molecules for it and you have the energy for it, there's a good chance that you might find life forms there. In Europa, we found something else. Europa is a moon of Jupiter, and what we realized when we looked deep, when we analyzed the last mission, we noticed that, first of all, in Europa, when you look at the ice cover is not old in Europa. Basically, you see a lot of new cracks. When you take photos years after years, you see cracks happening and the ice shifts. So there's some sort of a dynamics going below the ice that affects ice and produce new ice to come on top of the surface. So that's interesting in itself. But additional analysis of the magnetic fields that Jupiter affects all its moons and also on Europa also meant that there's probably a liquid ocean underneath, 10 to 15 kilometers of ice. And that's amazing. That's absolutely amazing. That means that it's probably salty because it's so cold and so far away from the sun. So to have a liquid ocean means that it has to be a lot of salts in it in order to lower the freezing point. And it also means that it's a really good applicant for life forms, because that thick of an ice basically allows for radiation protection. All the area around Jupiter is super radiated. It's really a harsh place to live. It's not a nice place to any life form whatsoever. So if you have a lot of ice and it's water ice, it's not dry ice. It's not the carbon dioxide. Then basically it means that the life within the ocean, liquid ocean, would have energy source and would have protection from radiation. And that's awesome. That's like a really, really good combination for us to find life forms.
Mm-hmm. Okay, perfect. We have another question by Sandeep Nirgude, but I guess this is asked by Sakshi. She's asking, in last session, you said that there used to be oceans of water on Mars. So what happened to them now?
Okay, so the process scientists now think happened on Mars was that it had a lot of ocean. It had an atmosphere. For some reason, its core, so planet Earth has an iron core, a molten iron core that turns around and, like a dynamo, produces an electromagnetic field around the planet. And this electromagnetic field really helps to deflect cosmic radiation particles that are coming from the sun. And these particles, because they are deflected by our planet, they don't bounce out of our atmosphere. They bounce off the electromagnetic field that directs their movement. In Mars, we don't know yet why. After about 1 billion years or so, we think the core stopped moving. The electromagnetic field stopped being produced because the dynamo was not working well. And all those particles from the sun and from other high radiation events in the galaxy started pounding the atmosphere of Mars. And what happened is that it's basically like putting like gamma radiation or even higher radiation on your atmosphere, and the molecules of the gases, they just break, they dissipate. And eventually a lot of the atmosphere just, blew off into space. And then you didn't have any atmosphere, so obviously the water started to boil up and to escape and to become vapor. But as they became vapor, there wasn't no atmosphere and no magnetic field to protect it and keep it as a vapor or go into a cycle of clouds or anything like that. So they also escaped into space and basically left the planet barren. Where we might find water ice on Mars today, and we do, is first of all in frozen form, and we know there is a lot of frozen form of water molecules on Mars, and those are in the polar areas, but also underneath some of the surface. And we're also looking for underground lakes, underground water reservoirs and things like that, and we haven't yet found one, I have to say that. But that's what happened to the water on Mars.
Okay, so actually we don't have many questions about the session that we have had today. I'm sure that Reut is really happy to answer the other questions as well. But if you have the questions related to this session, it'll be better. Anyway, we have few more questions, so we will take those. Adarsh Kumar is asking again, do we have any theory which explains how microorganisms survive on asteroids in the freezing voids of space?
Okay, the question, can you ask it again? Do we know what?
Basically, he's asking how the microorganisms can survive in cold environments on asteroids or in the space.
Yes, we do. So they go into hibernation and they don't have any water molecules in their bodies. So what happens, water, when they freeze, they expand. When water go into ice, they're actually expanding. So one of the things we realized very quickly is that if you are a living thing and you have 70% of your water is ice, and you have 70% of your body weight in water, liquid water, the blood and everything. And then all of a sudden you go into minus 80 degrees or minus, 200 Celsius degrees, and those water inside you freeze. They turn into ice crystal immediately. When they turn into ice crystal, they basically become very, very sharp. They become big, and they basically rip your tissues. Okay? So most organisms, what they do is that they make sure that they don't have any water molecules whatsoever inside their body, and they go into sort of like a hibernation. Because if you don't have any water molecules, proteins don't function. They just don't none of the system function in your body or in other microorganisms. So all the water are expelled usually, and then it's a hibernation. You can't really say it's a state of alive when someone is in hibernation, but that's how they survive.
Okay, perfect. So we have question from Parth. Can you explain central dogma of molecular biology in the context of beginning of life?
Can you explain the center dogma again?
Yeah. Can you explain the central dogma of molecular biology in the context of beginning of life?
I can't really. So aside from what I said before in the presentation, I don't know exactly how to talk about it. Can he clarify the question perhaps, or help me understand better?
We'll wait for him to clarify, and until then, maybe move on to other question until he replies back. Okay, so Aniket is asking, what provided the bonding energies to the molecules in primordial soup? Was it the underwater volcanic tubes, or was it some sort of external objects like meteorites?
I think the energy was provided. Again, it really depends what time in the beginning of Earth, whether it's the first billion or second billion years. But the sun is a huge energy source on planet Earth. Everything is basically on that's why until the 1970s, nobody thought anything is living underneath the ocean, because there's no sunlight there. So I would say that the sun, for most, was the most prominent energy source at that time.
Okay. We have another question by Adarsh Kumar, I guess. What's the possibility of organisms on planets like Kepler 22b to develop into Homo sapiens?
Very slim.
Yeah, of course. Like the way we discussed in the last session, we don't really know how the life would evolve on these planets, Kepler planets. There might be microorganisms or extremophiles, which we are going to talk about in upcoming lectures. So that might be it. But Homo sapiens would be like really thin. Okay, so Parth has explained his question further. He's asking RNA makes DNA, DNA makes RNA, and RNA makes proteins. So why didn't non-aromatic lipids and other... Okay, just a second. I'm getting confused in this. He has two questions.
Okay, where is that question? Is it up in the channel?
No. He's sending me on a different channel. So it's in my phone, actually. Yeah. Well, I'll ask you another question by him, because Parth, if you're hearing this, please explain your question in one text so that I can ask her properly. His another question is that why didn't non-aromatic lipids and other proteins form with the amino acids in Urey-Miller experiments both have similar organic structures?
No, they don't have similar molecular structures. So amino acids are pretty short compared to lipids. So if I understood correctly the question, so lipids have really long carbon tails, and they have a lot of hydrogen attached to it. While amino acid usually have one carbon, and it has 4 different groups attached to it, so it's a much shorter. So I think it's a good question, how does a lipid form in general? Like, how do you get that long tail with the hydrophilic head? Mm-hmm. And I don't know the answer to that. But for the amino acid in Urey-Miller, it was very clear that they're forming the short amino acid, as I explained, with just one carbon atom with different functional groups around it, with 4 different functional groups. Mm-hmm. That's what they found out when they did physical analysis on the racemic mixture that they got.
Okay, all right. So Parth is still typing. He's still making his question readable, but it's fine. Maybe we will answer it later in the next session or something like that.
Sure.
So I guess let's call it a session for now. We are coming back on 13th of September with another session, which is going to be about the evolution, the life and evolution. So I'll be posting the link in the comments of this talk so that you can go there and check it out and join in for the next Sunday. If you have any questions, do send it to us. You can email, you can send it on WhatsApp, you can send it on any social media, and we'll make sure that Reut gets it and she'll answer it in the next session for sure. Reut, this session has been amazing. Like, I didn't expect it to be so thorough and so it was really intriguing to even for me to know all these things. So I really enjoyed this session, and I'm sure that everybody who's watched this has also enjoyed, and we are absolutely looking forward to the other sessions as well. So I just wanted to tell you guys that we are going to have more of such sessions, not just by Reut. We had sessions with Ellen previously, and even after Reut, we are planning to have more sessions. So if you have any suggestions for us, do drop it in the comments so that we will know about what topics you want to listen to. And also, I am doing a session every Saturday. It's called Ask AstronEra. So I'll be answering your questions not related to this particular topic, but if you have any other topic in your mind, let me know. I'll surely answer those in Ask AstronEra. Also, if you comment on this video, we will make sure that you get a discount coupon to log on to our courses on astronera.org. So do subscribe to our channel, like this video, and put in a comment so that you'll get your own discount coupon for this. Thank you so much everybody for joining in. Thank you so much Reut, and we will see you guys on next Sunday at 6 p.m. Clear skies.
Clear skies. Thank you.